Hello and thank you for watching the video of my presentation. Before starting, I wanted to mention that some part of the data I'm going to present here are already covered in our previous meeting, so I won't spend a lot of time on them. I will try to highlight the more recent findings and make a connection with the previous data. That being said, this presentation will cover a very brief introduction to sodium pump, its isoforms, regulation mechanism, its function in skeletal muscle. Then I will present the major findings from 4 manuscripts I included in my dissertation. And if time allows, I will show some of the ongoing project and future directions. So sodium potassium ATPase is a plasma membrane bound enzyme or pump which uses one ATP, which hydrolyzes one ATP to pump 3 sodium out and pump 2 potassium into the cell. And by doing so, it establishes a steep ionic gradient across the cell membrane. By sodium potassium activity enables cell to concentrate ion. It also helps cell to have a constant ion environment. It regulates osmotic balance in the cell and prevents cell burst. And interestingly, in membrane cells, sodium pump activity enables cell to have a flexible membrane, and which eventually leads to the organism mobility. This is just part of a long list of cell-specific sodium pump function. The other function is that by establishing sodium and potassium gradient across membrane, sodium pump establish membrane potential. It also in neurons and muscle, the excitable cells like neuron and muscle, the pump activity preserve the excitability of the cell. And by establishing sodium and potassium gradient across membrane, it also provides a driving force for the transport of glucose, calcium, proton, amino acid, neurotransmitter, vitamins, and other molecules via second transport mechanism. Activity of pump also provide the energy to support the electrical signaling by action potential in muscle and nerve cells. So it's not surprising that 50 to 75% of total energy consumed in gray matter of brain, for example, are consumed only by sodium pump activity, and 25% of total energy in brain. So let's have a look at the sodium pump structure and subunit. Sodium pump comprise one alpha subunit here, one beta subunit, which is this one, and in some tissues, a tissue-specific regulatory protein from the family of fixed protein, this one. Sodium and potassium are transported by this catalytic alpha subunit. Beta subunit doesn't contribute to ion transport, but it's required for assembly, maturation, and function of pump. It's also essential for the translocation of the mature pump to the plasma membrane. The fixed subunit is not required for pump function, but it plays a major role in regulation of pump activity, and it's a target of adrenergic stimulation. Several isoforms of each subunit exist. Alpha subunit exists in 4 isoforms, alpha 1 to 4. Beta subunit exists in 3 isoforms, beta 1 to beta 3, and 7 different isoforms of fixed subunit has been identified. Studies in expression system shows that all 12 possible combination of alpha and beta subunit has distinct kinetic properties like different turnover rate, different potassium affinity, sodium affinity, and ATP ATP affinity as well. Since my project is about the physiological difference between alpha-1 and alpha-2 isoform, here I'm going to highlight some sequence similarities and differences between these two isoforms only. So within a species, it's been shown, it turns out that the sequence similarity among alpha-1, 2, and 3 is between 80 to 90% within a species. But between a species, the sequence similarity among alpha 1, 2, and 3 is at high 90%, which suggests that there is a strong selection pressure for expression of different sodium pump isoform in an organism. In terms of tissue expression of sodium pump isoform, alpha-1 is the one that is expressed ubiquitously in all animal cells, all cell types, and is the only isoform that is expressed in kidney. Alpha-2 predominantly expressed in skeletal muscle, heart, and brain. As for the subcellular distribution of alpha isoforms, in cells with highly specialized function and structure like neurons, heart, and skeletal muscle cells, isoforms of alpha isoforms are localized in different membrane domains. For example, in skeletal muscle, alpha-1 only found in the surface membrane, and alpha-2 is localized predominantly in T-tubular system and some other specialized domains like neuromuscular junction. So alpha-1 and alpha-2 isoforms of sodium pump have some evolutionary preserved differences in their sequences, function, tissue expression, and subcellular distribution. But what is the tissue-specific function of sodium pump in a skeletal muscle specifically? Skeletal muscle is the largest organ in human body with 40% of total body mass. It also contains the largest pool of sodium pump in the body, and most of the body potassium are stored within skeletal muscle, up to 70%. As for the contribution of sodium pump in muscle contraction, the pump activity is required for action potential. And during fight or flight response, the upregulation of pump activity helps to delay fatigue and keep us running. But muscle has other metabolic function like body potassium homeostasis. In this regard, sodium pumps of skeletal muscle tightly regulate body plasma potassium to prevent some dangerous conditions like low or high potassium level, which may lead to periodic paralysis or even death. So understanding the regulation of sodium pump in skeletal muscle is clinically very important because various malfunction in activity of sodium pump in skeletal muscle are attributed to several diseases like various types of muscular dystrophy, McArdle disease, and congestive heart failure. Skeletal muscle is also a good model if you are interested in studying sodium pump and especially sodium pump isoforms, because it's the only tissue that express a large amount of alpha-2 beside alpha-1 in distinct domains of the membrane. It also skeletal muscle also contain the most dynamic pool of sodium potassium ATPase, and the activity of sodium pump is regulated over a wide range. For example, from 5% of its total potential activity during rest up to 65% or more than 12 folds increase following electrical stimulation. The long-term goal of my study was to specify the role of different sodium pump isoform in skeletal muscle function. As a first step, I developed a method to quantify the sodium pump activity in intact myofibers isolated from skeletal muscle or primary myofibers. Before introducing the method, let's see how pump's activity is measured. So sodium pump hydrolyze ATP to pump out sodium, pump in potassium, and polarize the membrane. Therefore, it can be considered an enzyme or pump or charge transporter. To quantify the pump activity, we can measure the enzymatic activity by measuring the amount of ATP consumed or by the amount of sodium cleared from the cell or by the amount or rate of the potassium that is introduced to the cell, or by measuring the magnitude of negative charge generated in the membrane. But each of these methods has some interfering factors. For example, as in the case of a skeletal muscle, if we are going to measure the ATP consumed, there are other motor proteins that use a lot of ATP, like myosin. In order to get rid of that interfering factor, we have to homogenate muscle and purify membrane. In the case of measuring charge generated in membrane, there are other charge carriers, for example, sodium and potassium channels. So we have to block all sodium and potassium channels to be able to measure the specific charge generated by sodium potassium ATPase. And in the case of potassium increase intracellularly, there is a high potassium background inside the cell, which reduces signal to noise ratio. In this case, instead of using potassium, we use a potassium surrogate, rubidium, as a tracer for potassium. And for some experimental reasons that are beyond the scope of this presentation, this rubidium uptake method is considered a gold standard method of quantifying the pump activity. But even this method also has its own limitation that we described in this paper. And I introduced a new method for quantification of activity of the pump in a skeletal muscle. This is a mass spectrometry based method which use ICP-MS machine. Inductively coupled plasma mass spectrometry. I described isolation of primary myofibers from EDL muscle and show how we can measure rubidium besides other ions simultaneously by ICP-MS machine. And importantly, we showed that sulfur and phosphorus can be used as a normalizing factor for the heterogeneous samples in terms of cell volume or cell count. What are the significance of this method? In traditional uptake method, each muscle from animal provide a single data point. In new method based on myofiber, 100 myofibers from one mouse or like 2 muscle enable us to reduce the number of animals required for the experiment, and also provide a more homogeneous sample groups, which eventually increase the statistical power of the test. This is a mid-throughput method that is useful when testing the dose dependency of drug or a stimuli and its effect on the pump activity in a skeletal muscle fibers. ICP mass spectrometry method enabled us to use naturally abundant rubidium instead of radioactive isotopes of rubidium, which makes this assay safer, cheaper, and easily scalable. The next project that I'm going to discuss here is about the acute regulation of sodium pump isoforms by potassium concentration. But before that, let's review the general mechanism of sodium pump regulation. Sodium pump can be regulated at 3 levels. First, by by regulation of intrinsic activity of sodium pump, or how fast or slow the turnover rate of pump is. Second, by abundance of pump in plasma membrane. And third, by the total content of sodium pump in a skeletal muscle. So intrinsic activity of pump depends on the concentration of enzyme substrate, in this case sodium, potassium, and ATP, or depends on the protein-protein interaction, like interaction of pump with phospholamine protein. More on that later in this presentation. And also depends on the post-translational modification of pump, like phosphorylation or glutathione of different subunits. Abundance of pump in plasma membrane is controlled by translocation of subunit between subcellular compartment, like ER or surface membrane or T system. And the main mechanism to control the total pump content in a skeletal muscle is by translocation and transcriptional regulation of pump subunit expression, and to a lesser extent by regulation of translation of subunits and controlling protein stability and a genetic mechanism. In our 2015 JGP paper, we studied how pump concentration regulates the intrinsic activity of pump isoform, and we demonstrated that the potassium sensitivity of alpha-2 isoform is much less than what was previously suggested. In this paper, we measured pump activity by two methods. First, by measuring potassium sensitive currents in a voltage clamp myofiber isolated from flexor digitorum brevis or FDB from mouse foot, this muscle, and secondly by measuring the ATPase activity in muscle in mixed hind limb muscle from mouse foot. The mouse model that was used in this study was wild type and a skeletal muscle alpha-2 knockout, which is a targeted knockout of alpha-2 in a skeletal muscle, and the only active isoforms of sodium pump in the skeletal muscle of this animal is alpha-1. One major finding from this study was that in terms of potassium affinity of sodium pump isoform, K-half of alpha-1 is significantly smaller than K-half of alpha-2, which means that alpha-1 is more sensitive to more potassium sensitive than alpha-1. Alpha-1 is more potassium sensitive than alpha-2, and I will explain the physiological implication of this finding later in this presentation. In another related study published in 2018, we aim to obtain more detailed information on the dynamic effect of the plasma potassium on sodium pump isoforms. So we used different animal models, we enhanced our technique, and we further validated the specificity and sensitivity of our myofiber ICP-MS method. To measure potassium sensitivity of pump isoform, here we use a myofiber technique I developed, and myofiber were obtained from extensor digitorum longus muscle, this muscle from mouse foot. Tree mouse model was used, wild type skeletal muscle alpha-2 knockout, and swap. Let me explain what is swap mouse. So one significant difference between alpha-1 and alpha-2 isoform is their sensitivity to wobbling or dejectin, which is a specific pump inhibitor. Binding site is here. Alpha-2 is wobbling sensitive in wild type rodent, and alpha-1 is wobbling resistant in wild type. So with low concentration of wobbling, with treating sample with low concentration of wobbling, alpha-1 will be the only active isoform of the cell. This is very useful property when you are studying isoform, but the problem is that it allows the studying alpha 1 specifically, not alpha 2, because even low concentration of wobin eventually inhibits all alpha 2 isoform. This issue was resolved by introducing swap mouse by Dr. Lingrel and his lab. So in this mouse model, alpha 2 is wobin sensitive, alpha 1 is wobin resistant, and alpha 2 is wobin resistant, alpha 1 is wobin sensitive. So by treating samples with low concentration of wobin, you get alpha 2 as the only active isoform. And high concentration of wobin eventually blocks all isoform in all models, which is useful when separating specific versus non-specific transport activity. So we determined the specific potassium kinetics of sodium pump isoforms in a skeletal muscle by using myofibers from wild type alpha 2 knockout and swap mouse in combination with different concentration of wobin. Here we increased potassium and we measured pump activity, which gives us potassium affinity of pump. Wild type animal gives us potassium affinity of alpha 1 and alpha 2. Wild type mouse treated with low concentration of wobin gives us alpha 2 potassium affinity. Swap plus wobin, we have alpha 1 potassium affinity specifically. And another model, a skeletal muscle alpha 2 knockout, provide the potassium affinity of alpha 1 only. The results we obtain from all these three animal models are consistent, which suggests that potassium affinity of alpha 1 is significantly higher than alpha 2. Alpha 1 and alpha 2. And the maximum activity of alpha 1 is significantly lower than alpha 2, which probably is due to the lower membrane abundance of alpha 1 compared to alpha 2 in the skeletal muscle. What is the physiological implication of difference in potassium kinetics between these two isoforms? Let's go back to the localization of sodium pump in a skeletal in different membrane domains. As I mentioned before, alpha 1 expressed in surface membrane, alpha 2 predominantly in T-tubular system. The important thing is that muscle contraction requires a continuous train of action potential, and each action potential introduces a large amount of potassium into the interstitial space, here and also here. At the surface membrane, this extra potassium immediately diffuses to the plasma space. But at the T system, this extra potassium don't diffuse and is accumulated. It's accumulated. Why T system is diffusion limited system? Because of these extremely narrow T tubules, and because of the tortuosity, which is a feature of disordered network that is unlike ordered network, these systems are diffusion limited. So with K half of less than 1 millimole, alpha 1 is already saturated even at the resting extracellular potassium level of 4 millimole. There is no more room for upregulation. But as potassium is accumulated in the lumen of T system, it stimulates more alpha 2, and we can say that accumulation of potassium in T system regulates alpha 2 through stimulation and doesn't have any effect on alpha 1. This is consistent with the membrane localization of alpha 1 and alpha 2, and also with their potassium kinetics. My next project was about regulation of sodium pump by adrenergic stimulation. During exercise or fight or flight responses, all the mechanism that leads to fast and continuous muscle contraction are self-limiting processes. So a fixed pump activity similar to the resting level eventually impairs muscle contraction and leads to early fatigue. So we need maximum activity of pump to be able to run continuously. And this is the role of adrenergic stimulation, which is a reaction of sympathetic nervous system in response to a stress, to external stressor, by sudden rise of catecholamines, including epinephrine and norepinephrine. Both of them are shown to increase or decrease pump activity. But in the case of a skeletal muscle specifically, it's been suggested that the fatigue resistance effect of adrenergic hormones or neurotransmitters are through activation of sodium potassium ATPase. But the kinetics of action and the isoform specific target of this adrenergic effect is not known. In this manuscript, I did some muscle contraction experiment and I did some pump activity measurement to determine the kinetic and the potassium isoform target of adrenergic effect. But here what I did is that I electrically stimulated a whole muscle with 60 Hz frequency for 1 minute and recorded the time course of force decline. We calculated 3 contractile parameters from this recording, which are maximum force, time to half the maximum force, and area, the total area under the curve, which represent the total force generated over stimulation time. But the central question here is, what is the effect of adrenergic stimulation on muscle contractility? Or what is the effect of high extracellular potassium on muscle contractility? Is there any interaction between these two factors, high potassium and adrenergic stimulation? And what is the role of sodium pump isoform, alpha 1 or alpha 2, in response to these two types of stimulation? To separate the contribution of alpha 1 and alpha 2, I conducted this experiment in 12 different conditions. I had muscle from wild type and swap mouse, and I treated them with varied concentration of wabi. For the sake of this presentation, I used these blue bars to show when we had normal potassium, 5 millimole, or 10 millimole, high potassium stress, which is 10 millimolar potassium. And these red lines represent salbutamol, represent samples treated with salbutamol. Salbutamol is a synthetic form of beta-2 agonist, which is adrenergic stimulator. The gray line represent the baseline or no adrenergic stimulation, and this yellow window shows which isoform were active during experiment. So in wild type, the active isoform is alpha 1 and alpha 2. When we have normal extracellular potassium, the adrenergic stimulation has almost no noticeable effect. Compare this red versus green. When we treat a sample with high extracellular potassium, the treatment with high extracellular potassium itself impairs contractility. Compare this gray with this gray. And salbutamol treatment rescues contractility almost 100%. Then I treated wild type with low concentration of wabin, which gives us alpha 1 only. During normal extracellular potassium, salbutamol treatment slightly enhanced contractile profile, gray versus red. High extracellular potassium impairs contractility dramatically. this the effect of adrenergic stimulation is more pronounced here, almost 2 to 3 folds increase of total force generated following adrenergic stimulation. Compare gray versus red. When we have swap treated with low concentration of wobin, which gives us alpha 2, normal extracellular potassium has no effect, and adrenergic stimulation slightly enhanced contractility. High extracellular potassium, there is no noticeable effect of the high extracellular potassium when we have alpha 2 only, and also adrenergic stimulation has a very slight and non-significant effect on the muscle contractility. Compare red versus gray here. Here what we have is 3 different contractile parameters like peak generated force, area, total area under the force time curves, and the time to 50% of the maximum force generated. For example, here I indicated the peak generated force, low potassium, high potassium, and with or without adrenergic stimulation. Overall 12 different conditions. Similarly, for time to 50% of maximum force generated and total area under the force time curve. I've listed some of the noticeable findings from the ideal contractility experiment, but I just want to mention that overall our conclusion is that alpha 1, but not alpha 2, is the primary target of adrenergic stimulation. There are many confounding factors, there are many possible confounding factors that may involve in the effect of potassium and adrenergic stimulation on muscle contractility, because muscle contractility is a multifactorial, complex process. For example, two of those confounding factors are potassium-induced membrane depolarization and its effect on muscle contractility, or the selective effect of high potassium on alpha 2 specifically, which we mentioned before. To avoid these confounding factor and to reduce the effect of confounding factor, I measure sodium pump activity directly in 18 different experimental conditions in myofibers. What we have here is a rubidium uptake. It's not contraction anymore. Rubidium uptake by alpha 1, alpha 2, alpha 1 and alpha 2 and non-specific transporters. In high versus low potassium with or without salbutamol treatment. Let's look at each of them separately. So here we have alpha 1, and alpha 1 activity is upregulated by salbutamol regardless of potassium content. And extracellular potassium concentration does not have a big impact in pump activity when we have alpha 1. Conversely, in case of alpha 2 isoform, treatment with salbutamol does not have any effect on alpha 2 activity, but high potassium increased rubidium uptake. This is completely the reverse effect. And when we have alpha 1 and alpha 2, these contradictory effects probably cancel each other and we don't see a significant change. Another interesting effect of adrenergic stimulation studied in this project is that it changed the temporal dynamic of pump activity. Here we have time versus the activity of pump after adrenergic stimulation. In panel B, you see that salbutamol affects the time-dependent activity of alpha 1 and alpha 2 differently. This is alpha 1 and this is alpha 2. But in wild type, the adrenergic in panel A, the adrenergic stimulation just increase the activity of alpha-1, but doesn't have any effect on the time-dependent activity or the pattern of upregulation. But when I measured ATPase activity in purified membrane, not the whole muscle or not myofiber, I got somehow a contradictory result. Here, there is no considerable difference between alpha-2 and alpha-1 in terms of response to adrenergic stimulation. There are many explanations for this discrepancy we discussed in the manuscript, but the one that I want to mention here is related to the localization of alpha-1 and alpha-2 in the membrane. Skeletal muscle cells is a very large cell and is highly compartmentalized. So some of the cellular functions in skeletal muscle depends on the spatial access of one pool of protein to the other, to another compartment, another pool of protein, or another membrane domain. It's been suggested that in myofibers, the cyclic AMP, the core mechanism of adrenergic response, beta adrenergic response, the cyclic AMP production and PK activity are localized to different subcellular compartments. So it's possible that different adrenergic sensitivity of alpha-1 and alpha-2 that we discussed before is due to the asymmetric localization of this isoform, and therefore which determines the spatial access to signaling complexes and different receptor proteins. So most of the data I presented today suggests that there is difference between the role of alpha-1 and alpha-2 isoform of sodium pump. But the central question is why we might need this different isoform, or why several isoforms of same ion transporters are well preserved during the evolution, not just pump, other transporters. In terms of sodium pump isoforms specifically, the general consensus in the field is that alpha-1 plays a main housekeeping role, and alpha-2 or 3 are only activated during stress condition, or they involve in the fine-tuning and regulation of cellular function other than ion transport. This is a common pattern that is observed for many other protein isoforms. You knock out the isoform, you put organism under the normal condition, and often you don't see any effect, or the effect of the knockout is very mild. You put organism under a stress condition and the true function of isoform appears because the organism cannot tolerate this specific type of stress. In this view, isoforms is seen as a reserve capacity. But some of the results that I presented here, such as different potassium affinity, different roles of sodium pump isoforms in contractility and high potassium resistance, and also isoform specificity of adrenergic stimulation between alpha-1 and alpha-2, these results may imply that some isoforms, like sodium pump alpha-2 isoform, are evolved for distinct function and are not just a reserve capacity for the stress condition. I don't think that I have time to cover the clinical significance of the malfunction of isoforms, and I would like to finish my presentation here. Thank you very much for watching this presentation.